The overall goal of this procedure is to quantify the proteolytic activity of botulinum neurotoxin in complex matrices such as pharmaceutical, environmental, and food samples. This is accomplished by first processing test and standard curve samples, if necessary, to establish suitable pH and viscosity conditions. Next, the botulinum neurotoxin is immunoprecipitated from the samples using antibody-coated magnetic beads. Then, the magnetic beads are thoroughly washed to remove any interfering compounds and resuspended in an optimized reaction buffer. Finally, the washed beads are incubated with a protein reporter and spectroscopically measure the cleavage of the reporter over time. Ultimately, comparison of reporter cleavage in the test samples versus the standard curve samples is used to quantify the activity of the botulinum toxin in the test samples with mouse to near-mouse bioassay sensitivity.
The main advantages of this technique over other methods, such as the mouse bioassay, immunologic, and other fluorescent methods, are that this assay does not require the use of animals and has been demonstrated for use in highly complex matrices found in food, environmental, and pharmaceutical samples. Generally, individuals new to this method may struggle because careful sample processing and dilution is required. Demonstrating this procedure will be Dr. Mark Dunning, a scientist at BioSentinel.
Prepare buffers according to the text protocol. To generate a standard curve for quantifying test samples, weigh out 10 plus or minus 0.01 grams of solid food sample into a 50 milliliter conical tube and set aside. This sample will be used as the diluent.